Hello, good evening, everyone, and welcome to tonight's webinar. This is the first in our series all about genetics and regeneration. And we have got Dr. Gustavo Torres de Souza leading this series, happening every Wednesday in July. And he's going to be guiding us through different topics in genetics and helping you decide if this is something that you want to introduce to your patients and look into testing options and the different things that we can consider. So it's going to be really exciting, really innovative talks. And to get started, it'd be great to find out a little bit about you and what your current experience level is. We're going to pop some polls up on the screen now. So if you can tell us what your medical profession is, please. And there we go. We've got these rolling in, and I know lots of people are just joining us. So we're just trying to get a little bit of background information on who's here this evening. So yeah, 40% doctors, 20% dentists, 20% nurses, 20% other. Okay, and next question should be appearing on the screen now. Okay, should be popping up any second. And this is just to give us a little bit of info on what your experience, if you've got any experience in genetic testing yet, bear with us. And it comes up. No, not quite yet. Should be here any second. Nope, this is sorry, still the same one, guys. We should have it coming up in a minute. Let's see, is this it? There we go, understanding genetics. So how confident are you in your understanding of genetics? Would you rate yourself as a beginner level, intermediate level, or advanced level? And if you can just pop in your answers. There we go. Okay, so majority here are classing yourselves as intermediate. So that should be a good help for Gustavo leading tonight's presentation. And then the third question should be coming up any minute. So have you ever used genetic testing to inform your treatment decisions? A majority are saying no. Okay, great. You're in the right place, guys. So Gustavo is a pharmacist. He holds a doctorate in genetics and has extensive experience in genome editing and the generation of genetically modified animals. So he's currently scientific director for Fagron Genomics and specializes in pharmacogenetics. So he's well versed to guide us through this evening. So I'm now going to hand over to Gustavo. We can get started. If you do have any questions, then please pop them in the Q&A box below, and we'll answer them all at the end. And we aim to wrap up by 8 o'clock this evening. So there we are. Over to you, Gustavo. Let's get going.
Thank you very much, Chloe. And well, thank you everyone to be present. Well, as Chloe has already presented me, I'm Dr. Gustavo Torres, and the aim here is for us to start discussing genetics. We'll have plenty of time this month to have all these webinars. So obviously, again, please let me know now in the question and answers, but also in the course of the month, if you have any specific questions, anything specific you'd want to see in the course of these webinars, just please let me know so we can go on improving and also bring in more content to you. Also, obviously we'll be doing a question and answers in the end, but if anything pops in for you, if anything interesting or important appears for you, I'll try to also follow the chat so I can answer live if anything very important appears or anything that needs to be addressed during one of the questions or one of the slides. So I will share my screen at the moment so we can start talking. So, well, the webinar series is going to be a 5 webinar series. So the first one we're going to be dealing today is the principles of genetics applied to regenerative medicine. And we have four more coming, which are going to deal with other aspects of genetics. So to clarify, today we are basically going to be dealing with the DNA side of the genetics. So might sound silly just to say, well, DNA as genetics, but there are other macromolecules which are important and are interesting for us to understand in view of regenerative medicine, so as to say. So this is the aim of the moment, really, is to see all the extent of molecular biology and genetics and how it interacts with regenerative medicine. So we will be dealing with genetic testing, we'll be dealing with predispositions and so on. However, our aim here is to provide you with a a large amount of information and wide information on the use of genetics and the understanding of molecular biology in view of regenerative medicine in general. So for the first webinar, as I've said, we are dealing mainly with DNA. And this is the table of content for today. Here today, we essentially will be dealing with the molecules in general we have and how we come from DNA to, the other molecules we'll be talking about, such as the proteins, essentially. And then we move on to the DNA as a center. Sorry, I just need to check something. And then we'll be dealing with DNA per se as a source of information, both for cellular functioning and for cell function and for information in the body, in the functioning of cells in general. Um, and then we move on to the use of DNA and the machinery, the DNA replication machinery that is involved in replication, repair, and genetic variations as well. So this is where we are starting to get to the side of genetic testing and genetic variability in patients. And finally, we'll talk about a bit, not about the use necessarily of genetics as a tool, but the knowledge in genetics that help us understand the translational part of regenerative medicine. So let us get started. These are I've sent out with the Institute personnel people some references you can use to guide you through the course and also to find further information. For this first webinar and for all the other ones, there's this book in the beginning. There is the Griffith Introduction to Genetic Analysis. This one I can't share with you, obviously. But also the information is also in two papers I've shared with you and also in the material. So also in the course of the week, in the course of the month, please let me know if you need information or if you need any additional details into the references, or if you need more references to understand or to add any information for you. And so we can start with what we're looking at. So instead of simply telling you that I'm going to go through the basic topics of genetics or the basic topics of molecular biology as a simple thing. What I really want in the course of this month, especially because we have a considerable time, is to really go into the hard, the difficult topics so that you can actually understand how this impacts your clinical practice. So the aim here really is to provide you with good information and deep information in genetics. So as you all know, obviously, regenerative medicine is built upon biological components. So we aim at alterations in the tissues, the cells, so cell proliferation, tissue improvement, and overall body body alterations. However, when we want to understand or to apply clinically these principles or the principles in regenerative medicine and the clinical application of it, we need to understand first that we have a molecular basis to any of the treatments you might be devising, any of the treatments you might be giving to the patients. And this always is an interplay between macromolecules that are going to have a structural property. So they might be proteins, they might not be proteins. Um, we have cell signaling. So this is mostly going to be proteins interacting either with small molecules or between among themselves. And we have the cell properties answering to either uh, the environment and this structure we have, or the response to proteins. So this brings us to the aim, or the final aim, or the first webinar, but also of the series, which is to help us understand how this interplay happens and what are the most important components and how they actually work in the deep end of the science, which is genetics and molecular biology. So for us to start, we have many both claims and uses in the clinical practice in view of nucleotides, of peptides, other macromolecules. And it always is important, at least from my side, I see that in the clinical practice, it is important for us to get a better understanding on what we have exactly, so what chemically actually we have when we are talking about this type of molecules. So nucleotide are the building blocks of our DNA and RNA in our body. So the functioning of our body, we do have DNA and RNA. But also, if we go to the therapeutic side and we have the polynucleotide, the PDRNs are of sorts, they also are chemically exactly the same. So they are exactly the same thing chemically. So we might need to understand DNA and our RNA and nucleotides in our bodies first, and then we take a look at how they might function therapeutically. And the same thing goes for amino acids, which are the building blocks for the peptides and proteins we use therapeutically, but also obviously are the same are the same peptides and proteins we have functioning also in our bodies, and they have their function, their structure, their correlations, and so on and so forth. So as a take-home message from this beginning is that, well, we do have product names, we do have product structures, and all of them are going to be correlated eventually to a cell function that we want to address, we want to improve in the patient. So when we start with nucleotides, there's a fair bit amount of new products in view of nucleotides. So a few new products which are nucleotide based, and I think it's important for us to start by understanding, or start um, um, by discussing how um, how nucleotides function, so as to say. So I think that is obviously everyone knows what is a nucleotide and what are their uses, so as to say. However, I don't think everyone has visibility on the structure of nucleotides per se, and this is what I want to us to look at. We're going to look at nucleotides more in depth because this class is going to be essentially for DNA. However, the first thing I'd like you to see, and this is majorly the main application of the polynucleotides, although sometimes it might not sound as such. So sometimes you might think that, the signaling functions might be important. However, one of the main importances, um, um, or the main information we need to know, or we need to have when we're talking about polynucleotide therapy, would be really the use of the polynucleotides as a form of hydration. So they actually do have this chemical property. So the DNA per se does have a part of the informational property. It also does have chemically a property of binding to water. So this is going to be the first and foremost information with regards to are polynucleotides, which is the fact that we do have in the phosphate part of the DNA the capacity of these hydrogen bonds forming between the water molecules, but also between the water molecules and the oxygen atoms that we have in the backbone of the DNA. So if we look at the DNA like that, we know that whenever we have DNA, either in its normal format, in its double helix format, or when we have it broken, we always will have a group of water molecules binding and hydrating this molecule all across it. And as we have, a water binding to it very strongly, which is the hydrogen bond, it's a very important intermolecular bond. What we will have is that we have the possibility of using this molecule as hydration. So the first and foremost information we have related to nucleotides is that it is useful for hydration. So it will always connect to water and will always keep water. So this is the first information, is that it chemically is also it also binds to water. As for when we go to amino acids, which are going to be essentially what we have as building blocks to peptides and proteins. We have a different structure because obviously they are different molecules, they are different macromolecules, and they are bound by the peptide bonds. So peptide bonds. So this would be one amino acid, and they are bound by this kind of bond, which is essentially the peptide bond. What is important about the amino acids is that apart from the fact that it does do a secondary structure, which is this initial conformation that we get from the bindings between, or from the bonds between the amino acid residues inside the peptide, we move on to having more possibilities of structure. DNA has many possibilities of structures, however, amino acids has more. And why does it has more, or the reason why amino acids are so interesting and peptides are so interesting, is that as we have more possibilities of binding, we also have the possibilities of having peptides binding and forming proteins and looking like this, which you all will recognize as the collagen fiber. So it looks like a fiber, so it's simply going to be a long structure and it stacks together. We also have the possibility of more complex, like that, structures being formed with the proteins. And as more structured and more complex structures are formed, we have a second level of, or we have a final level of complexity in these molecules that leads us to what we have on the left, which is the possibility to form receptors in our bodies and interact either with big molecules or small molecules. But what is important here is that, for instance, we have proteins forming this kind of structure, which is a very complex structure that passes through the membrane and brings information from outside of the cell to inside of the cell by changing the configuration. So let's say here we have a small molecule binding to this receptor, and as it binds, it transfers information from outside of the cell to inside of it. And what is important here, we have a final kind of confirmation of the protein molecules that leads it to being functional in the sense of having more variability of functions, of having the possibility of signaling to a cell, altering the state of a cell, and so on and so forth. So this is essentially what we look at when we want to talk about a protein at its final level, which is essentially a protein that has a structure outside of the cell and inside of the cell. And when we have something binding to it, so let's say we have a small molecule that comes here and binds to this protein, it does generate a signaling that goes to inside the cell. So you see that the fact that we have proteins or amino acids are forming these proteins that are able to make these tridimensional configurations, that leads us to the possibility that as these proteins fold and unfold, they also create the possibility of getting information to the cells and altering the states of the cells. And therefore, we have, before we go on then to actually the hard stuff in DNA part, we do have as information here, or as a summary here, the fact that when we are talking about applying a certain therapy to a patient, we have to think about before starting that we have a type of molecule, we have, and within this type of molecule, we obviously have things like peptides, proteins per se, like big proteins, growth factors, which might be proteins, nucleotides, vesicles, and so on and so forth. What is the kind of signaling it does to the cells? There might be a vehicle, so it might be being carried in a formulation, and what is going to be the final tissue response. So as you see, we begin from the small molecule and we go on to which tissue response we expect to see in that patient, and this is going to be obviously highly connected to the molecule we have in the beginning. As a summary, of these two types of molecules. So we have nucleotide-based therapies and peptide-based therapies. Although we're going to discuss a bit more nucleotides today because this is the DNA day. Um, although this is important, um, although it is important to also note that there are more types of response to nucleotides. Uh, The primary function of nucleotides in the regenerative medicine is the fact that structurally it does become bound to water and it does lead to hydration. So this is a first look at the nucleotides. While when we go to peptide-based therapy, so protein-based therapy, what we have is the possibility of a certain peptide, as we have, for instance, here in our body, the GLP-1, but also externally, so also outside of our body and used as a drug. We have other peptides, which are, for instance, semaglutide and so on and so forth, tirzepatide, retatrutide. What happens is that they have a physical tridimensional structure that binds to the receptors, which are also proteins, and they have this capacity of transferring information from outside the cell to inside the cell by changing this configuration. Well, so as a summary here, we have as nucleotide derived, we have DNA therapies, RNA therapies, which are newer therapies, as in gene therapies. We have then the polynucleotides, PDRNs, of sorts used in cosmetics and regeneration. So we do have here three main mechanisms, and one of them is the hydrophilicity, the nucleotide salvage, and the signaling potential, which we're going to discuss after we talk about DNA. When we talk about peptides, growth factors, cytokines, collagen, we have here firstly the possibility of signaling per se, so actually folding receptors and binding to the structure, and we also have the possibility of adding structure to a certain tissue, which would be the case of collagen, for instance, although obviously we don't use it very much physically in the format of final form of collagen. Well, and then finally, it takes us to the DNA part of our session, which is obviously going to be my favorite part of it. When we talk about DNA, it is important for us to start discussing when and how we had um, DNA being introduced as a science or genetics being introduced as a science. So we had initially Mendel, so you certainly do remember from uh, from school, who started identifying hereditary patterns in plants. So he used to breed peas, and he noticed that there should be something, there should be a factor inside the cells that lead um, the final individuals to present a certain characteristic. And this was the beginning of genetics per se. Later, we had many more additions, and one of them is not very well known, but it was done by Griffith, and it is this experiment on your right. And this experiment is something very interesting here because we are talking about DNA in about its structure and so on. However, when we start talking about DNA and genetics as a basis and also as a basis for patient variability and so on, which is also going to be a topic for us during these webinars, uh, we need to understand DNA as a source of information. So what happens here is that in this experiment, it's going to explain a bit kind of a very basic experiment that was done back in the days in which we had um, a pathogenic strain of bacteria, which is this one, the S strain, and we had a strain of bacteria that was not pathogenic, which is this one. And as you can see, we always go on injecting an extract of this strain that is pathogenic and live bacteria. So this means we are destroying bacteria up here, and we are putting it in contact with the ones we have here. So when we attempted not to destroy anything or to destroy polysaccharides, lipids, RNAs, and protein, it always happened that when we merged it to the live bacteria, it always got some characteristic that still led the mice to die, as we can see here. However, when we removed DNA from this extract of bacteria we had here in the beginning, so when DNA was destroyed, the mice would live. What does it mean? It means for us that DNA has something, so the molecule of DNA, it has something that transfers information. So we know DNA and we use DNA in practice as a molecule for its properties, physical properties and chemical properties. However, DNA has inherently also the property of transferring information. And this was essentially what started to be the center of genetic analysis and the center of genetic information from then on. So this was when we started to understand genetics better from the view of DNA and use DNA as a source of information. So today we're going to go from DNA as a source of information to again back to DNA and peptides as a source of therapy for their characteristics, for their chemical structure. So we will begin again with DNA. So now we're going to go back to the beginning of DNA as a source of information and also as a source of and also as capable of uh, um, allowing cell renewal, cell rejuvenation, cell proliferation. And then we go back again to using this as technologies. So as I started to mention back in the days, Mendel had noticed that if we had peas of different colors and we bred them, so this was a true experiment. Uh, we saw patterns in how this appeared across the offspring, so as to say, of this pea. What is important here is the fact that Mendel had noticed that there were factors which he didn't know to be DNA yet. He didn't know to be genes yet, but there were factors inside this individuals or these individuals that led the characteristic or the physical characteristics to be visible and to be different between them. And this was the most important aspect of Mendel's work, and also what really helped us understand what was genetics in general. So it was the fact that there was something inside the cells that led to information being transferred from, the the initial individuals to their offspring. And how is this information initially structured chemically? So chemically, what we have in the DNA is that the nucleotides are the basis for the DNA, and they also are the monomers, so they essentially are the single units that form the polynucleotides. And DNA obviously is one polynucleotide. What we have is that we have in our DNA at least, it varies a bit in some very specific species. We have four different types of nucleotides. So we have four different types of monomers, which are adenine, guanine, cytosine, and thymine. We're going to discuss this a bit better and how they bind, but the most important part is that we have a phosphate part, we have a sugar that is a desoxyribose sugar, and therefore this is why it's named DNA, and we have a nitrogenous base. As we have seen in the beginning, but also here with a bit more detail, we have the phosphate part binding from one nucleotide to the other, so from one residue of nucleotide to the other. And as you can see, we have here many oxygen atoms outstanding from this part. And we also have this in this side, we also have obviously a hydroxyl here. What is important here is that this is the part, so this binding in the DNA we see here and we see here, so the phosphate part, this is what is responsible for water to being able to bind here. So the hydrogen bond that allows DNA, allows polynucleotide as a structure, as a molecule, as a therapeutic molecule to be used for hydration is essentially this part. So water molecules are capable of binding here due to the characteristics of these oxygen atoms. So the phosphate part of the DNA is going to be responsible for the hydration we get from using polynucleotides. Also, obviously, in the structure of DNA itself, which is going to be important for its function in our body, this is going to be kind of the backbone of the DNA. Well, as well as DNA is structured uh, chemically as a linear molecule, the information contained and the information we have from this connection between the adenine, thymine, cytosine, guanine, it also allows us to have information being transferred as we pair DNA. And a second important thing about the DNA structure is that when we leave the structure, the chemical structure, to the informational structure, what we have is that we have parts of the DNA that are responsible for different things. So we have here different combinations of these letters or these ATCGs that are responsible for regulating the function of the DNA. We have some combinations that could be this one that interact with proteins such as the RNA polymerase that is responsible for producing RNA, and we have proper portions of the DNA that have different combinations of these letters that led us also to coding for proteins in the future, which is going to be one of our topics. So essentially, this allows us to see DNA biologically. So the function of DNA inside our cells, it starts with the fact that DNA replicates itself and it has information. This information is transcribed to RNA and it's then translated to protein. So we have already seen from the chemical and therapeutic side the use of DNA as a polynucleotide and the proteins as a peptide. But in our bodies, these molecules also do carry functions. And these functions, they do overlap a bit with what we use in terms of therapeutic use of both polynucleotides and proteins. So what you're seeing in your screen now is described as the central dogma of molecular biology, which describes the flow of information from DNA to protein. So this linear flow of information is what essentially um, shows us that we keep information and we replicate this information in our cells in the form of DNA. So from one side we have DNA, and on the other side, what we have is protein as a main um, factor or as a main um, executor, so as to say, of the biological functions. In the middle of that, what we have is RNA. So from DNA to protein, what we have is always the fact that we have DNA here in the cell nucleus. Just going to write it instead of drawing. And DNA is responsible then, we know already that DNA is responsible for storing information. However, what happens is that how this information is actually delivered to a body or functions in our bodies, that we have RNA as an intermediate. So RNA is produced here in red, as you can see, um, being translated, being transduced, being transcribed from DNA, and then it leaves the nucleus to the cytoplasm in order to produce proteins. So proteins are then produced in the cytoplasm. And this is a kind of a last view or more detailed view in the production of proteins. So in this production of proteins, what we know is that we have this correspondence between these three letters at a time, which is called a codon, to one specific amino acid. And this is how we go on building proteins in our body. Well, and what is important? We start with DNA, storing information, we produce proteins, and we have many different types of proteins. So the output of the DNA information is in most situations going to be proteins, and proteins have many different types of structures, as we have seen, and therefore they also have many different types of functions. So we have proteins being entirely structural, such as we have for the collagen, actin, tubulin, keratin, and so on. But we also have proteins that are hormones, so small proteins, which are peptides such as insulin, but also immunoglobulins, and so on and so forth. So the important part of proteins is that as they become more complex, they have more complex structures, and also they are able to produce more types of functions in our body. Well, as you can see here on your right, um, the fact that we have growth factors interacting with the cells is another way of looking at peptides and growth factors that we use in the clinical practice as proteins. So let's say we have these smaller proteins, and they are able to interact both at the same time with the structural part of the tissue, which is also proteins, and also cell receptors transferring information to inside the cells. And this interplay between cells producing one certain type of peptides, which could be growth factors, and signaling to other cells, this is going to be the so-called cell signaling that is very important in the therapeutic use of proteins. So as a final message here before we go on to the DNA as renewal, we have the fact that regenerative outcomes, so the use of these molecules in regeneration, they don't come simply from um, one necessarily structure of a certain molecule. So let's say the polynucleotides, the peptides, and so on, but they do come from this interplay between how these molecules interact with the cells and the cells interact with one another. And biologically, so inside our body, what we have is that DNA stores information, RNA is the intermediate needed to produce proteins, and then we have tissue response and alterations in the body. However, there's an interplay between what is happening in our body, so meaning the DNA replication, production of RNA, production of protein, and proteins that come from the outside, or polynucleotides that come from the outside therapeutically, and are able to interact with the system. So this is essentially what I want to show you in the course of this month, is the fact that we have things happening in our body, so in the cellular machinery we have, that do interact dynamically with what we are introducing as the therapeutic factors, let's say as molecules we are adding um, in in a therapy proposal to a patient. And that takes us to the renewal part. So we talk too much, and we see and we hear too much in the practice of um, in the practice of regenerative medicine. We hear too much about renewal, cell regeneration, and so on and so forth. Um, However, there are inherent biological factors that are happening inside our cells and in the interplay between our cells and tissues that do lead to this renewal to happen per se. And also there are, cellular machinery that are needed in order to happen, or in order for our certain tissue to recover. What do I mean by that? Let's say we have a certain injury. So we had an injury here. What happens is that we don't have a simple one type of cell just simply proliferating and generating new tissue there. What happens is that we have several types of cells proliferating and communicating between them, between types of cells and among types of cells and the proper tissue per se. And therefore, regeneration in the sense of here simply remodeling the tissue that has been injured, it does need cells to interact inside themselves, so they need to proliferate, but they also need to interact with the tissue and among cells. So this means that cells need to divide in order to renew rejuvenation to happen, but it also needs to happen in a conservative and controlled way. And this is essentially what happens in the cell cycle, in which we have DNA replication happening prior to cells being able to separate this DNA and divide them in two cells. So whenever we talk about this, it's important for us to understand that, well, the DNA replication we're going to see in the middle, because this is the DNA webinar, it does happen in this part of the cell cycle, but it also requires several stop signals that can happen between each one of these mechanisms, are essentially because they are able to say, well, this cell is proper, is good to continue dividing, this cell is not good to continue dividing. So before cell division, um, before a cell needs or can divide, what we need to happen is the fact that DNA inside these cells, it needs to be duplicated. So it needs to be copied. So let's say we have one cell here, and before we have two cells, DNA needs to be replicated in these structures we're going to see. And how this happens has a very important impact in how rejuvenation and how cell renewal and in general, our regeneration happens in our body. So um, replication per se, I'm going to describe how it happens mechanistically, but there's a second most important part to it, uh, um, which is essentially the interplay of the control of this DNA replication. So DNA replication happens when cells are about to divide and it has signal from outside to divide. So bear that in mind. And what happens is that we start by having proteins that are going to unwind the DNA and separate the two strands. After that, we have the helicases keeping the DNA separate. And then finally, we are able to have the DNA polymerase attaching to the DNA and starting to bring new nucleotides to producing more DNA. So these are the kind of main steps. We have DNA being separate, we have the helicase organizing DNA, and then we have the DNA polymerase entering right here, and it goes on polymerizing the DNA in our body from one side and to the other side, happening in steps. So what happens here is that DNA replication is very deeply linked to cell renewal and to tissue regeneration. So clinically, what happens is that we need cells to start, when we need cells to divide, they need to know they can divide. So they also need to know that there's some external signal or some internal signal to that cell that leads a certain protein to recognize an origin in the DNA. So the origin is going to be where this process is going to begin in several parts of the DNA we have in our cells. Most importantly, in order for tissues to renew, we need this to happen, and we need the signaling to happen from outside the cell to inside the cell. So this is majorly going to be done by proteins. And as this happens, what is important for us is that we have the possibility of controlling proliferation. So the control of proliferation is very important, and also the fact that DNA needs and must be replicated before we start producing new cells. So as a summary here, and also as a very important aspect in the clinical use of regeneration, and what we expect when we have active ingredients that will lead cells to proliferate and to promote regeneration, is the fact that regeneration, as we understand, is not simply cells dividing, but rather the fact that we have a control in cell division. So in each one of these steps, we have a very strict control so that cell proliferation happens after DNA is able to divide. So after DNA is able to duplicate so that we can keep information. So the effectiveness of tissue repair, so the use of tissue repair, it does require cells to divide properly and in a controlled way. We have a last part here in the DNA part before we go on briefly to the last clinical comments, is the fact that this is going to be discussed later. But one critical part in DNA proliferation, in cell proliferation and DNA duplication, is the fact that we have in the tips of the chromosomes, so this is also DNA, we have some repetitions called the telomere. So we have some repetitions in these letters that are called telomeres. And as cells divide, the telomeres need a slightly different structure to be duplicated. And when it happens, the telomeres we're not able in all cells to keep the telomeres the same size. What happens is that we go on losing bits of the telomere. So the rest of the DNA is mostly informational and so on, while the telomeres are kind of in the tips of the chromosomes. And as we go on losing them, cells might go towards the side of senescence, or also we might have malignant transformations. Why is this important? This is important because this is one of the major actors in regeneration that connect the cell division to the cell senescence. So cell division and how cells continue to divide or not is in one side majorly controlled by the fact that the telomeres control the the cell's capacity of keeping the DNA integrity and stability as well. So as we lose too much telomere, we would expect cells to start or to stop dividing, but also we might have problems in this process. We're going to discuss telomeres in another time, but just so you bear in mind that telomeres are also DNA and they also have this connection. As a last topic in DNA per se, before we go into the final remarks, DNA specifically as we have discussed, it is linked to carrying information. Uh, so as DNA carries information, this is a topic we're going to discuss along the month or across the month. However, as DNA carries information, it is expected that DNA carries information slightly differently between individuals. And this differences are what makes this different. So the difference in the DNA is what makes patients differently, but also what makes them respond differently to therapy and so on and so forth. And one of the major players in DNA variations are what we call SNPs. So this is going to be topics for later, but I also wanted you to know that a SNP is this alteration in one single letter. So we have a CG in this individual, and we have a TA in this other copy of the DNA. So this is going to lead to alterations in how this portion of the DNA is going to be read by the cell machinery, and later it's going to influence in how this is going to respond. Or say, that means these patients are going to respond to a certain therapy, or how they're going to evolve clinically. So, as a summary of the DNA part, we have DNA as a molecule. Um, and as a molecule, it does store biological information, and therefore it will define cell function. But also structurally, DNA also might lead to hydration. So when we use it as a technology, it also will lead to hydration. As a functional output, we have DNA producing or being used to produce RNA and in the end protein. So it does have tissue function. However, technologically, we also can use peptides to produce cell function and to alter cell function. DNA biologically needs to be duplicated in cell division, so DNA also is important to the controlled cell renewal and regeneration. And finally, biologically, DNA is the source of clinical variability across patients. So these are the four main important things for us to know about DNA. And then we go finally to the translational part, some last slides, and I want to discuss them with you more closely. What is important here is the fact that keeping in mind DNA, when we start a treatment in a certain patient, it is important to know what is being used clinically. So what type of molecule we have, what are the mechanisms and target cells these molecules might target and elicit in terms of alterations, which tissue processes will happen, and essentially what evidence we have. When we talk about polynucleotides, I've talked to you about the fact that, the very structure of DNA, so as you can see here again, the backbone of the DNA, it does correlate to simply uh, hydration. However, the fact that we have DNA also having this tridimensional structure, when it enters the body, we do have some other possibilities of use, or other possibilities of mechanisms in the use of polynucleotides. Um, let's just recall something here. When we're talking about tissue regeneration and wound healing, which are going to be things we are going to use uh, um, as an example here, we have the fact that cells need to populate this area here that has been wounded, for instance. However, as you can see here, while cells repopulate this, we not only have proteins being produced, but we also have a structure of DNA, free DNA being put there. So bear in mind that this might also be one of the main mechanisms by which polynucleotides and PDNRNs they work in the bodies, is that from one side, we might have more DNA available structurally around a certain tissue, as you can see here. So it might be occupying the tissue and providing nucleotides or free nucleotides to the tissue to allow the tissue to recover. And this is one of the potential mechanisms. So the salvage pathway is one of the mechanisms we have for DNA or for polynucleotides to be used therapeutically, which comes from the PDRN degradation. And as a second mechanism, or potentially a signaling mechanism, is the response in the A2A or the adenosine receptors. Because when we have DNA or the nucleotides becoming available, as you can see here, they might bind to the adenosine receptors and elicit stall control. So as I've said in the beginning, we always have an interplay between what we have externally being introduced in the patient and what we have internally happening in our cells. So from one side, we have in the salvage pathway the fact that for tissue recovery, we need cells to proliferate in a controlled way. Um, and for this proliferation to happen, we also need DNA duplication. And for DNA duplication to happen, we need polynucleotides. So this polynucleotide we have injected in the patient, it might go to this pathway and provide nucleotides to the patient's recovery. And from the other side, we also have separate nucleotides binding to the adenosine receptors, and the adenosine receptors might lead to cell functions which already exist. And these functions, they are majorly led by proteins transferring information from these nucleotides to inside the cell, leading to controlled inflammation, cell proliferation and migration, and alterations in the tissue matrix. For us to finalize the polynucleotide part of the translational side of the use of DNA, we have the fact that and this was reviewed by someone you surely know from Rami and Ramsey. So what you see here is essentially the fact that polynucleotides, they have different mechanisms, as we have seen, um, and it needs to be further studied. However, it aligns with a biological logic of repair, angiogenesis, inflammation control, and cell proliferation, and also matrix reorganization. And they'll all lead us to understanding that we have a plausible biological mechanism to know that PDRNs are polynucleotides. They are able to promote hydration, and this alters the structure of the tissue. It allows tissue to regenerate because it makes available nucleotides, and also they might signal to cells through the adenosine receptors to promote cell proliferation, organization of the tissue, and control of inflammation. So, uh, the aim I had here in this session, essentially, just so we can finalize and go to questions, is the fact that we have from one side one biological molecule, which is the DNA, and this biological molecule, it leads to several mechanisms. We have biological mechanisms, and we have the very structure of the DNA being able to interact with how the cells function, and in the end, how tissues are renewed and recovered in the end. So I would like to thank you all for your presence and open for the questions and also further discussions that we might have um, either now or in the course of the following weeks.
Thank you so much, Gustavo. That was an excellent presentation and a great start to our series on genetics and regeneration. Hopefully, everyone is really geared up now for the next few weeks and has got all of that basic information and understanding of DNA there. So, does anyone have any particular questions that they would like to ask Gustavo now? Then please pop them in the Q&A box at the bottom of the screen. Um, one of the main things that I would like to ask is if there's one thing that people should take away from this evening, what would it be?
I have a question before, which would be essentially who uses nucleotides as therapy here? Please happen. Please go on and answer. So, who of you has already used our nucleotides in therapies?
There we are. If anyone wants to pop that in the chat. let us know what you're using. That would be really good. Is everyone feeling a bit shy? Because we've still got a few people online. Be good to find out a little bit about what you're using.
Well, guys, so I will discuss a little bit further. So in the next a few weeks, we will keep discussing these basic mechanisms and their connection to the very uses of molecules and macromolecules are involved in genetics, um, in rejuvenation, cell renewal, and regeneration in general. And I would like you all to take back home the information that essentially what we need to understand is that there's always a cell mechanism and there's always a basis in the functioning of our own molecules that lead these therapies we have to functioning. So what I want to discuss with you in this series is essentially the basis so that you all have a very good information, a set of information in the back of your mind for whenever a certain new therapy starts to be developed, start to be offered. I want you all to know, how these therapies really work and how they really interact and will help your patients so that that the therapies can be better thought, better discussed, and um, we can provide better care to patients. So again,
Absolutely.
thank you all very much.
Thank you. We have just had one question come in, Gustavo. Does overstimulation shorten the telomere?
I would say that yes. So for most of what we know, overstimulation to cell proliferation, it will shorten the telomere for the simple fact that cells will be dividing faster. Um, we have different we have different types of cells and different types of let's say, types of cells doing proliferation. So if we are able to provide or to promote cell division, so if we are using an active or using a certain technique that leads to more cell proliferation, and many of the energy-based devices, the physical devices such as microneedling, injections, and so on, they are aimed at cell proliferation. So certainly we know that there's going to be some degree of stress to these cells, and there's going to be some degree of um, telomere loss in these cells. So this is for sure. We are not sure yet how this correlates clinically, so how clinically this is going to impact the overall regeneration or the overall regenerative aims we have in these techniques. However, whenever a technique really does stimulate more cell production, there's always going to be some additional stress to these cells, and there might be some telomere loss. And overall, in the cells we have in the adult body, they all have this, decreased capacity of keeping the telomeres. So we always will be losing telomeres to a certain extent.
Thank you very much for that. And then somebody has said, thank you so much for making such a complex topic understandable, which is brilliant.
Thank
Thank
you. Thank you very much, Dr.
you for that.
DeJoya. Thank you.
Thank you so much. And we'll continue every Wednesday now throughout this month. So join us here, 7 o'clock UK time. As you've already registered for this, you'll be automatically registered into the continued sessions. You'll just get the login information sent to you next week. And also bear in mind our conference Ramsey is tickets are available, early bird tickets are available until the end of this month. So if you want to chat to Gustavo there, meet in person, want to learn more about regenerative aesthetics, then do join us there and get your tickets while they're discounted. So head to the Ramsey website for that.
And
But for now,
obviously, these topics are going to appear in the Ramsey event. So, take the opportunity for us to kind of start discussing them, having a basis so that then in the Ramsey event we can go even deeper and you can go even deeper
mhm.
in all the sessions you'll have there because in the end all the regeneration is based in biology. So this
Yeah,
is what my aim here is for you.
Exactly. Thank you so much. And everyone, don't
Thank
forget
you.
about the learning that Gustavo sent over. There's information in that pack that was downloaded from the email for the rest of the sessions. Just so do have a look at that beforehand. Thanks, guys. Thank you so much for everyone's
Thank you.
time.
Thank you all very much.
Bye.
Bye bye.